everyone. Thank you so much for joining us today. Before we officially kick this off, I do want to point out to the audience that we welcome questions. Please post your questions in the Q&A section at the bottom of your screen, and we will address those questions at the end of today's presentation. So I'd now like to turn this over to Steven Saltzstein, Force Family Office's CEO.
Thank you, Callie, and thank you everybody for joining us today. We greatly appreciate you taking time out of your schedule to be with us. We had a family office breakfast in Miami yesterday, and we had about 25 families. And as we were going around the room, the sense you were getting is that this market is the worst of times and the best of times. And what I would say is it seems to be the worst of times for the legacy tech companies. And the feeling was it's the best of times for biotech. And if you take a look at Phio, and this is just my personal opinion, if Phio was private, it would be worth hundreds of millions of dollars. But because it's public, there is this kind of embedded discount. And our families absolutely expressed yesterday that they're looking for undervalued growth opportunities, and they are looking at life sciences as kind of their number one target. So with that said, it is my great pleasure to welcome Robert Bitterman, CEO of Phio. Thank you, Rob.
Thank you very much, Stephen. And I'd like to thank everyone who's in attendance, giving me a little of their time so that I can tell you the Phio story. So Phio is a clinical stage biopharmaceutical company, and we specialize in treating skin cancer. And what's very important, I think, to understand is that we do this with a very unique technology, which in the form of a drug has the ability to make our immune cells more effective in killing cancer cells. And so with that, let me just spend a second showing you our forward-looking statements, since we will probably get into some forward-looking comments. And let me start with what I believe is the foundation of the Phio value proposition. The first of which I think is the key point that we are targeting the second largest incidence of solid tumors in the human body. There's 1.8 million incidences of a condition called cutaneous squamous cell carcinoma. It is an unsatisfied market based on the various treatment approaches that are available today. And an accessible market value in this total area could approach $20 billion. The second feature of the value proposition is that we're doing this with a unique paradigm-breaking technology called INTASYL. And what we're doing this is for skin cancer, but it can also be applicable in other forms of solid tumors as well. And again, we use our body's own immune cells to more effectively kill cancer cells. And we're doing this based on the results of our first large clinical study. We're doing it safely and very effectively. And I'm going to get into showing you the results of that study in a few minutes. The third point I want to raise here is that the technology that we have, INTASYL, is broadly patented out through the year 2040. And it's not just a single patent. There's combinations of composition of matter, use patents, and formulation patents. So it's very strongly patented in a number of markets around the world. Finally, we're bringing this to you with a group of 8 employees and 4 subject matter experts who are part-time contractors. And except for 3 people in the financial area who are relatively new to the company, all of us have worked with each other through various companies in the field of dermatology for a number of years together. And we basically, myself, as well as my 2 senior colleagues, we actually came out of retirement in order to take on this program because we're so excited about the technology as well as its application for medical improvement in this area. And just the fact that it has the opportunity to generate a significant amount of revenue and profit generation and return on investment for our shareholders. So what is this all about? Well, Phio has a history of actually innovation. It starts with our co-founder, a gentleman named Craig Mello, who actually won the Nobel Prize for his discovery of RNA interference. And that discovery ultimately led to the development of our current technology portfolio called INTASYL, which is a short interfering RNA drug technology. And what's unique about this is that it has the ability to precisely silence specific designated genes in the human genome and in effect making our body's immune cells, our T cells, more effective in killing tumor cells. So how does this work and what's the rationale? By selectively silencing the signal from a designated gene, we can suppress protein production from that gene, which in the presence of various tumors can cause our immune cells to become totally dormant and ineffective. And so what we're doing is by suppressing that protein in the presence of tumor cells, we can reactivate the body's T cells and make them go into a role where they can actually do their job to attack foreign invaders in the form of tumors. What does it look like? I'm not going to spend much time on this, but I'm going to give you the non-scientist's point of perspective and reference on this. Simply think of this drug as two strands of a number of very carefully sequenced fragments of RNA. And what do they do? The sequencing of these little fragments gives exceptional target specificity. What I mean is that we can basically target a gene to silence that signal and not hit anything that we don't want to hit. And in addition, we can do this by protecting the stability of the drug and allowing the drug to actually be injected through the cell membrane of a tumor and get into where it has to be deposited without, remaining it intact and doing it through a concept called endocytosis, which is nothing more than the ability of the drug being sucked in to the gene or the cell and doing its job that way. A better picture of how it works. And once we get past this, we'll move on to some more mundane data. But if we look at the left side of the screen and we see a ribbon-like substance, that's a cell membrane for a tumor. And the squiggly material going through it is the drug. And so I told you about those two strings in this construction of the drug. The one string, which is the passenger strand, guides that drug across the membrane intact into this little blob called RISC. And RISC is a RNAi-induced silencing complex. The passenger strand leaves this other strand, the guide strand, and this is where the action takes place. It attracts the messenger RNA, the gene that's giving the signal, and it basically connects to it, and it basically, through an action like metabolic scissors, slices it, severs it, cuts off the message, and the protein is stopped, allowing the drug to, or the gene and the T cells then, to actually do their job and kill the cancer cells. It's that mechanism and work that helpfully has created over 59 issued patents with a number of additional ones pending out through the year 2044. And as I mentioned earlier, the composition of these patents, it deals with the chemistry. We also have different patents on the drug compounds that we can create from that, even on specific gene targets, as well as on therapeutic indications. And so in the old days, when this company was just a discovery company, the scientists were rather prolific. They created a number of different compounds, which you can see here, and I'm not going to go through them. But when I was asked to come on board and take the reins in an operating capacity, we concluded that we can't do all these. We have to find one or maybe two that would be most effective. And what is that going to be? What's the criteria for that? We had to determine if it could be safe in man, that it could be medically meaningful, and if it could satisfy those two, could we actually get an attractive return on investment for our shareholders? And so with that, we said we will focus on a select program or two, and the rest of them, which are non-strategic, we will try to monetize in an out-license program. And so we started with this drug we call it PH762, which actually silences a particular gene called the PD-1 gene. And I'd like to give you a couple of thoughts as to why we chose that one, as opposed to all those other ones that are on the list. And the first reason is that as a small company with limited bandwidth of people and cash, the first thing I was concerned about was risk mitigation. How do we make sure that we're basically going to direct our activities in the right place and mitigate the amount of risk that could be associated with development? We knew that PD-1 had already been validated by the large pharma companies in their monoclonal antibody studies, and they had done so in skin cancer trials. But they were doing it very differently. They were attacking the PD-1 gene on the surface of the tumor cell by infusing the drug throughout the entire body. We knew that in our preclinical studies, we could inject the tumor and that we could basically turn off PD-1, not on the surface of the tumor, but actually right into the T cell itself, turning off the supply of PD-1 like a faucet and never allowing it to escape to the tumor cell. With that, we then looked at what was the opportunity for market size. And when we looked at cutaneous squamous cell, we did see that it was a significantly large market and that additionally, the number of players in the competitive landscape was fairly limited. And so this seemed like a pretty good idea in terms of where we'd be able to direct our initial attention. Here's just a quick example. You can see cutaneous squamous cell in this pie chart, and it dwarfs all the other solid tumors in the body, which gives you a sense of the size of what the market we were looking at. So with that, we filed an investigational new drug with FDA back in 2023. And with that, the FDA cleared us and allowed us to study late-stage melanoma, late-stage Merkel cell, these are stages 4, late-stage cutaneous squamous cell. But with one caveat, they said, we'd also like you to study stages 1 and 2 of cutaneous squamous cell carcinoma. And that really changed the whole dynamics of the economic model for us at Phio, because over 70% of the cutaneous squamous cell tumors are in stages 1 and 2. And these are tumors that have a size of 3 centimeters or less in length. The other interesting point here is that there is no currently approved FDA drug to treat stages 1 and 2 of cutaneous squamous cell. The standard of care is surgical intervention. And while surgical intervention is actually very viable, it has its limitations. Depending on the size of the tumor, the location of where the tumor is, the patient's general health, there's often a need for a non-surgical option. And maybe a more visceral way of describing this would be if you're going to be subjected to different forms of skin surgery, how big of a hole do you actually want dug into your skin? And what's the likelihood you're going to be able to have that repaired through plastic surgery, through skin grafts, and suffer with all the things that go along with the recovery period? And so it was therein that we saw the opportunity for 762 and intratumoral injection of the drug as a means of therapy. So we embarked on a clinical study, and I'll tell you just briefly what the scope of the study was. We was designed to study up to 24 patients in 5 dose escalating concentrations, meaning the FDA was looking to see at what point could we find a level of concentration of drug that actually would basically be a dose-limiting toxicity, as they call it. Well, we studied 22 patients that completed treatment. We gave them 4 intratumoral injections over a 3-week period of time. And at the end of 5 weeks, if there was any tumor left, it was resected. But we took a biopsy to find out and measure the pathology in terms of the tumor results. Is there any cancer left? We examined the safety at each examination point. And we also looked at pharmacokinetic analysis as well. This study was completed in January, a month ago, and it was done through a number of prestigious sites. So what I'm going to do now is tell you about the very specifics of what came out of that study. And the outcome of this is really, it's kind of multifaceted because we not only had very interesting efficacy, but we had superb safety, we have a convenience factor, and there's some interesting economics associated with this as well. So let's first start with the clinical efficacy. At week 5, we determined that of 20 patients that specifically had cutaneous squamous cell carcinoma, of those 20, 14 of them were responders. And I'm going to tell you what a responder is. A responder is very specifically defined as one of 3 categories. The first category is, do you have 100% removal of cancer? Is it 100% clear? That's one form of response. The second form of response was, do you have greater than 90% clearance, or are you left with less than 10% cancer? And the third was, do you have greater than 50% clearance? And, or in other words, you have less than 50% cancer left. Well, in this particular case, 14 of the 20 patients were responders, and 10 of those 14 were 100% clear. That means that the pathology revealed that there was no cancer remaining at week 5. 2 of them at week 5 had greater than 90% clearance, meaning there was less than 10% cancer left, and 2 of them had greater than 50% clearance. There were 6 patients who did not respond, but those patients actually did not also experience progression of the disease either, so they were stabilized. Okay, so that's the efficacy, and these numbers are actually quite remarkable for a study of this nature, because skin cancer in some of the other trials in the monoclonal antibodies does not respond to the same rate. But the other interesting factor here is the safety profile. We increased the dose 20-fold, 20 times greater from cohort 1 to cohort 5 in these patients. And in not one case did a patient receive a dose-limiting toxicity. The study, by all practical purposes, is very safe. And the increased dose, the highest dose, was actually the highest concentration that we could physically put into the buffered saline, which takes the drug actually through the cell membrane to the target. Why is that? Well, I think we have to say that the direct injection into the tumor essentially eliminates off-target side effects typically associated with monoclonal antibodies, which are systemically infused throughout the entire body in order to reach the PD-1 on the surface of the tumor cell. And when systemic infusion of monoclonals occurs, the serious adverse event rate is somewhere in the range of 37%. So that's a significant challenge from a safety point of view. And these take the form of various autoimmune problems such as myocarditis, different problems in the respiratory system, and also in the colorectal system as well. The other point on safety is that we're not injecting a large quantity of drug either. It's going a small quantity being carried with buffered saline solution, nothing else. Many drugs, in order to get across the cell membrane, they got to put formulation enhancers in there, and they create their own form of local toxicities. We don't have any of that. We have no lipid nanoparticles. We have no viral vectors. And our treatment procedure is basically time limited. It's not a permanent change. It's just done long enough so that the T cells can recover their activity, reinvigorate, and go after the cancer cells. Next point is interesting is convenience, because this drug can be injected by a physician in a physician's office. This avoids the logistics of systemic infusion centers where one has to make appointments, where the doctor may lose direct control of the patient in a different setting. It also, INTASYL minimizes the aftercare associated with surgery, with those individuals that you often see walking around with bandages on their face and other areas. That's a function of the surgical intervention that they've had to endure. And lastly, but not unimportantly, we can, based on the fact that different lesions come in different sizes, we have the ability to flexible dose these based on the patient's individual characteristics and the configuration of the package. Another factor in this is interesting is economics, because with this driving patients into the physician's office, it drives patient practice economics because the doctor is paid for the procedure to inject. On a broader sense, we're talking about basic solid phase chemistry synthesis, which yields a relatively low cost of goods. And just as a noteworthy point, the source of our active ingredient, as well as the drug product itself, are manufactured in the United States, which eliminates concerns that people may have regarding tariffs or variable pricing that would become unattractive. So how do we position this? Well, we look at this based on what we know today and think of this drug as, at a minimum, tissue sparing, meaning that if we can't eliminate that tumor, we're going to shrink it. And if we can shrink it, we'll minimize the surgical excision, we'll minimize plastic surgery afterwards, preserve skin integrity, promote faster healing, and reduce pain. And we've already talked about the safety profile. We could not find a dose-limiting toxicity in the highest dose that we delivered, so it appears that the drug is favorably safe. And finally, it offers the convenience of being able to send the patient to the doctor's office, and again, also provides an economic benefit to the injecting physician as well. So that's the story on 762. I'd like to just spend a quick moment and talk about the other program that's in our portfolio, which is called PH894, and it targets a different gene called the BRD4 gene. And this program has been in a pre-IND filing stage for a while because we want to take the majority of our focus and our dollars to basically keep our eye on the 762 exercise and make sure that we don't get distracted. But this is a very interesting program that at some point when the financial condition is appropriate, we would like to move forward on this. It is a gene, BRD4, which is implicated in various types of skin cancers throughout the body, or not just skin cancers, but every type of cancer throughout the body. And our drug is specifically and precisely selective just for BRD4. And this is important because other companies, large pharma companies, have tried to study to treat this with small molecule pharmaceuticals, and they could not isolate BRD4, resulting in off-target side effects that became very toxic and caused discontinuation of the study. But in our program, we have demonstrated to date that in a non-human primate, that we have a very clean tox profile. It's also an interesting drug because it works in two ways. It not only kills the tumor, but it, like 762, it activates the immune cells to make them more invigorated to go after the tumors as well. And as I mentioned, this is a program where the IND-enabling studies have essentially been completed, but we're going to have to wait until the funding comes in to basically take this to the next stage. So let me talk briefly about the metrics in terms of where we stand today. Well, actually, as of 12/31/25, we have 11.6 million shares outstanding. There are 13 million plus warrants that have a conversion price of $2 a share and have a short life of less than 2 years now. They have an exercisable cash value of approximately $26 million. We don't carry debt. Our head count at December 31st was 7. Today, as I mentioned earlier, it's at 8. We added another person in our development group along with our subject matter experts. And so that basically gives you the core outline of what we're about on some metrics. And I guess this will take me to summarizing and say, Phio Pharmaceuticals is a fascinating company driven by a broadly patented technology called INTASYL, which has the ability to precisely silence genes in the human body, and a number of them. The opportunities extend far beyond what we as a company today can afford to do, but it offers opportunities for other companies who may want to license or get involved in the technology down the road. And our team is validated by their track record. As I said, we have worked together for many years and as a group. So we collectively know how we work. We've done this. We're into this because we want to do it. We're excited about it. It's not because we have to do this at this stage of our life. And finally, the market size just for cutaneous oncology is significant. The addressable market could exceed $20 billion. And so with that, I would just leave you with the thought that we have the ability to make our immune cells more effective in killing cancer cells, and I will open it to your questions.
Great. Thank you so much. And let's get into the audience questions. So is this a one product company, or does this technology have the potential beyond skin cancer?
This technology can address a number of cancers. And in fact, the PD-1 gene, which we can silence, is implicated in head and neck cancer. It is also affected in liver cancer and several other, not prostate, I'm sorry, but a couple of other cancers as well. That's just the PD-1 gene. The BRD4 gene, which we can silence with our other compound, which I talked about as being in a pre-IND stage, that has the ability to concentrate on a variety of cancers: breast, liver, also head and neck, melanoma, uh, uh, pancreatic, I believe, a number. So this is not a one-trick pony. This thing can be developed as far as one has the resources to do so.
Great. Thank you. In simple terms, out of the patients treated to date, how many saw their tumors significantly shrink or disappear?
Of well, there were 22 patients treated in total. 20 of them were cutaneous squamous cell. Let me add that one patient was a Merkel cell, one patient was a melanoma. Those were harder to come by for the simple reason that those patients had to fail on prior therapies before they were allowed to go into our drug. Okay. When we talk about shrinking, essentially we're talking about pathology. We're talking not necessarily the size of the tumor at the time we looked at it. We're talking about the amount of microbiological cancer that's left, which is actually a much more important factor. But in that regard, 14 of the 20 squamous cell patients had a response that ranged anywhere from 100% down to greater than 50% reduction in cancer. One Merkel cell patient miraculously had a greater than 50% response rate, which is a very deadly disease. And that person actually got a response, and subsequently, with some additional surgery, we understand, and an additional infusion of an antibody, actually has gone in remission. The melanoma patient did not respond, but also the melanoma patient was in the earliest phase, which had the lowest dose of the drug as well. So 14 out of 20 responses. Of the 14, 10 had 100% complete response.
Wow, great. Thank you. And what is the average age of patients?
I don't know exactly the age of the patients that were in this trial, but by individual, but I can tell you that essentially I'm going to speculate that almost all of them were at least 65 years of age or older. So this is a therapy that oftentimes is going to be affected by Medicare, if that's where the question might be going. But yeah, these appear in the later stages of life, and usually people in their 60s. But it can appear sooner, but that seems to be where the trend is.
And would this initially be used by dermatologists, cancer specialists, or surgeons? And how easy would it be for them to adopt?
I think initially, well, first of all, this is a relatively new type of therapy because injecting tumors has not been that common in the past. This plays very well into the hands of a derm surgeon, uh, more so than an oncologist. Certain advanced cancers are more likely to be treated by an oncologist and sending them to infusion centers for monoclonal antibody treatment. So we suspect that the target will be derm surgeons. We think that the approach will be, and this is something I didn't mention in the presentation, but the stages 1 and 2, if left untreated, are going to advance to stages 3 and 4. So this is not, insignificant. And the other point is that the rate of mortality on cutaneous squamous cell is as high as melanoma. That's something that's not well known. But one of the reasons is because there's so many more patients, and if they're not treated in the earlier stages, they will advance, and then it becomes a very, very complicating problem for them. But I think this is best suited for the derm surgeon, either as a direct line to treat patients based on where their lesions are. For example, at the risk of getting too detailed here, lesions on the lower limbs do not heal. And so that's one place where the surgeons do not want to cut because the problem of basically healing either with skin grafts or plastic surgery oftentimes is not successful. So it can be complementary. It can also be a first-line therapy. That's how we see it.
Since surgery is the standard today, who is the treatment clearly better for, and who is it probably not right for?
If we're looking we that's a fair question. It's clearly better for the patient who has early stage cutaneous squamous cell. And obviously that's where we've got the majority of our results to date. And we've seen the effect of the tumor shrinkage or clearance. So for a patient who is prefers to have 4 injections over 3 weeks as opposed to having it cut out, uh, it's probably a preference for the patient. A lot of people have, if they've already had surgery, derm surgery in the past, many people come in that it's not the first time. They've had multiple surgical procedures, and I've had one myself, which was pretty mild, but I've talked to people who have actually found the procedure to be quite discomforting and painful, and cosmetically an issue as well. So um, this is probably, going to be somewhat patient directed in terms of what their preference is. If you want to get a fast cut, the doctor can probably cut that thing out, but then you're going to have a longer recovery probably as well. Um, but at this point, what we don't know is how effective the drug will be in the later stages of things like melanoma and Merkel cell. We got that one result, but there's not enough information there to tell us. This could be certainly an important adjunct therapy to the monoclonal therapy as well as a booster. Uh, that's another aspect of this. There are many things that are being explored in terms of our curiosity, but we can't generate the funds to basically find all these things out. And this is where it could be very useful for a partner who wants to come in and look at some other aspect, a different disease state or whatever, that they could look at this.
Well, you bring up another question is, are you looking to partner with a larger company or, to become acquired? And what kind of progress would you need to make this realistic?
Very interesting question. And the larger companies, first, I we suspect that this product will probably have the most interest to companies that are in and around the dermatological fringe of things. Okay. And right now, this is a very innovative therapy that most people haven't, in the larger companies, haven't quite come to grips with. They don't understand it. But many of the dermatologically related companies, they're into other types of small molecule therapy. I personally think that this is an excellent addition to a larger company's portfolio. Now, we're talking about very huge companies like Merck, uh, Novartis, Regeneron, companies of that nature. There is, at the risk of being a little cynical, when it comes to looking at things that are not made in-house, oftentimes pharma has been known, and I've quoted this, I'm quoting one of the largest CROs in the world saying they're the last to be first and the first to be last. And I think because of that, uh, they're not going to even basically take a look at this until it's further along into the next stage, which takes us to probably the question, what are we going to do next? Which I'm prepared to answer that question if somebody asks it. But I think we're a little bit we don't have quite enough money, not money. We don't have enough money to do all the things we like, but we certainly don't have enough information to attract the huge players. And you also have to understand that those companies. I've worked in as a small business unit within Aventis Pharma before it was acquired by Sanofi. I know the political dynamics in the companies between business development and research and development and how people go about choosing things. It's a complicated process that you really have to be in the right place at the right time, and the pharma company has to be in the right mood. But I think a mid-sized player could have a big interest in this. And the answer to, are we looking to we would like to take this clearly to the next stage because we believe, Steve alluded to this, that the value is really understated based on what the public market is dealing with us today. And it would be unfair to our existing investors to not realize much more value out of this, which I believe we can get. if we move it to the next into the next pivotal phase here in the studies. So I don't want to hog that other question, but the question may come up as to what we're going to do next. So
Right. And is the drug manufactured for a particular patient?
not specific, no. It's just designed to basically shut down a specific protein generated by a gene that is in everybody's body. And it just so happens that when PD-1 is present as a protein in the face of tumor cells, T cells basically, shut down. They go dormant. You remove the PD-1 and the T cells awaken.
And what specific milestones should investors watch for over the next year that would tell us the program is working as planned?
The next milestone that we will be dealing with is compiling all of the data in this study. We know what the top line results are and the safety, but for purposes of FDA submission, our next step is to basically approach the FDA and say, this is what we would like to study next. This is the approach. This is the statistical plan. This is the number of patients that we deem are appropriate from additional safety. And this is the progression. In fact, our goal is to basically go into what is called a phase 2B with a rollover into phase 3 or a pivotal trial and bypass the old traditional phase 2 totally. We believe that the data is compelling enough based on the efficacy as well as the safety profile that we have a very good chance of getting that possibility. So we'll be submitting a package to FDA sometime before the end of March, which will have far more detail in it than what we've shared here today, which will basically boil this down about lesion size, what cohort the patients were in, what gender was the patient, in far more detail than most of us would want to know. But that's what our clinical group and our statisticians and our regulatory people are working on now. We'll submit that, and we'll be requesting to do a phase 2B rollover into a pivotal, which would mean that that pivotal trial would then be the trial that would be examined by FDA at its conclusion for an NDA file acceptance. And there are a few of that. So that March submission, I think we're going to say that we're doing that. Okay. Is that a milestone? We'll report on it, of course, when we do it. The more important element of it will be, but that's a significant step. The other important element will be, what is the FDA's response to that when they take a look at that sometime or preferably the early part of June. That way we will know exactly how many patients that the next trial is going to require, whether or not they buy the concept of taking phase 2B rolling into a pivotal. That will be a major step. And there's one other step in between that we will attempt to take a shot at. We don't know if we'll be able to get it, but we will attempt to see whether or not that this drug qualifies as a breakthrough technology. And we will request that that'll be independent of the filing with the agency for the guidance meeting to see whether or not they'll entertain that. We'll find out. But that's the intention, to do that sometime, I believe, around the end of this month, the early part of March, and see if we get breakthrough designation, which would be a great thing to have. That's gives us a pathway to better relationship with the agency and probably facilitates the whole pathway to approval.
Okay. Thank you. At the current spending rate, how far does your cash take you? And will you need to raise more money before key results are available?
The current cash will take us well into the first quarter of next year. As to key results, it depends on what you define as key results. If you're talking about the pivotal trial results, yes, because the monies that are available right now do not cover the cost of what would be the phase 2B3 study. That's a study that could range anywhere, and this is based on advice that we're getting and based on different points of view. That patient trial is probably going to it could be anywhere between 120 to 160 patients, and we're assuming that the cost of that trial probably about $100,000 per patient. So we're talking somewhere in the range of 12 to 15, $16 million out of pocket to conduct that study. So we will have to raise money for that.
Okay, great. Well, that concludes the questions. For the audience, if you have additional questions or want to be connected, please reach out to me. Everyone has my email address. Excuse me. In turn, Phio Pharmaceuticals will have your information and will be reaching out as well. And Robert, I do want to point out to you, back to you, any final words before we close out today?
Well, first, just to thank the attendees for giving us some time to present the story. And just a passing suggestion, skin cancer affects a lot of people, and many people don't realize it until it's in the later stages, and then it becomes very complicated. So my suggestion would be, if you are spending time outdoors in the sun, I would suggest a skin cancer checkup with a dermatologist once a year. It's better to find out and to be safe than sorry, because the longer it goes, the worse it gets. So just a friendly suggestion.
Great. Well, thank you so much. And thank you everyone for joining, and have a great day.